everyone. This is Yamina from Dr. GPCR, and welcome to the video edition of the Dr. GPCR newsletter for February 2023. And today I am delighted to have with me as my co-host Anne-Marie Quinn. She is the CEO of Montana Molecular. Montana Molecular is one of our ecosystem partners, and I'm super excited to have you with us, Anne-Marie.
Oh, thanks, Yamina. It's great to be here.
Thank you so much for taking the time. This is a new format for the news that we're exploring. It's more fun than reading a newsletter, watching a video, and I'm excited that we're doing it together this afternoon.
Great. Looking forward to it.
All right, so where do we start? We've talked before hitting record about the fact that there's a couple of news coming out of Montana Molecular. Also great news with Dr. GPCR, the ecosystem, and Montana Molecular. So maybe we should start there.
Yeah, I mean, you're right. There is a lot of good news coming up, and I guess for us, one of the big events coming up is the annual meeting of the Biophysical Society next week in San Diego, and then that's bookended following right after with Society for Laboratory Automation and Screening. So it's exciting for us because we have not traveled since February of 2020, and this will be our first time out, and hopefully everybody will stay healthy. But I'm excited because we are kind of teaming up with Dr. GPCR with our promotion. And we have gifts for people that come by that are branded, both Montana Molecular with Dr. GPCR. So T-shirts and coffee mugs and I don't know what all else, but yeah, it'll be fun. I'm really looking forward to that.
I'm excited about that. Can you please tell us what are the dates specifically for the SLAS meeting?
Well, SLAS ends on March 1st, and then I can count back from there to 28, 27. I believe it begins on the 25th with the pre-meeting short courses.
Yes.
And one thing that's neat is another friend of ours is doing a short course. Do you know, I'm sure you know Terry Knocken.
Of course.
So he's doing a short course, and Sam Hoare from Farm Mechanics and also Montana Molecular, he'll be joining us. And he's presenting two posters with one that talks about the use of biosensors and kinetic signaling analysis that he's developed, and another one using like what the value is of doing live cell time course assays for drug discovery and how that really can inform the compound profiling and getting better data early on in that discovery process. So I'm really excited to be there.
That's exciting. I hope he's not presenting both posters the same day.
Well, if he is, I'll probably bail him out and present one of them. I don't think we really have our times yet, but I think what's neat about SLAS is you go on Sunday and put up your posters and they just stay there for the duration. Yeah. It's not like you have that little 2-hour window like some of the more academic conferences. So
yeah. I was at SLAS last year in Boston, and it was really neat. I just went for the day, but it was Disneyland for scientists, which is amazing.
Really fun. And we're unusual because we're an assay development and services company, but so many of the exhibitors there are instrumentation, robotics, and just great new things. I mean, I saw Eppendorf and Agilent both have new instrumentation out there. So it's always a great time and great to see people there. And I just feel so remote from that for having stayed home for 3 years, But I'm excited too because we are up for the new product of the year award. So that's exciting, and that's for Borealis, which is our new biosensor for beta-arrestin recruitment. And we had launched a prototype, maybe right sometime during the pandemic, a prototype arrest and biosensor. But in the last 6 months or so, we've just really evolved that biosensor to be brighter and have a better signal to noise. And so we can hit a lot more of the receptors now with that assay. So we're excited
Exciting.
to present that. And what's neat about it is having a biosensor for, say, uh, beta arrestin, as well as our caddis sensor for cyclic AMP, or as well as our assay for diacylglycerol. You compare those, and you're making a measurement with using the same assay modality. So it's not like you kind of have to figure out, how do I compare this calcium dye with an endpoint arrestin assay? You've got the same assay modality. You're measuring it with the same analytic tools, and being able to extract similar parameters and very easily compare how signaling changes based on either the receptor or based on the compound. So
That's amazing. I've never had the chance to use in the lab any of the Montana molecular tools, but I always found them very neat. And I love the idea of being able to measure multiple things at the same time in the kinetic fashion, and also having the ability to measure, you're transducing the cell, so you can use basically virtually any cell that is transducible. And so you're not constrained to your typical CHO or hex cell system.
Yeah. We're actually getting started up, getting some neuron cultures into the lab, some DRGs and cortical neurons, and starting up some experiments doing neuronal cultures. And we a lot of us in the lab, not me personally, but several of us in the lab come from that neuroscience background. So there's a lot of excitement about getting in those primary cultures and just taking them for a ride.
That's amazing. That's really lovely. Before we move forward, I just want to take a moment to thank you and Montana Molecular for being our ecosystem partners. The company has supported us since day one, since the first Dr. GPCR Summit in 2020, and we're very grateful for the friendship and the support. So we're very excited to see you receive the prize, and I can't wait to see the data. That's okay. These things happen. These things happen. I want to take a moment also to thank our ecosystem partners, Domain Therapeutics, GPCR Therapeutics, Design Pharmaceuticals, and our newest addition to the Dr. GPCR Ecosystem family, Orion Biotech. And some of these companies, including representatives from Montana Molecular, are going to be next week in Boston at the second GPCR's Targeted Drug Discovery Summit. We're also partnering with the event, so I'll be there, and I'm very excited about it. And before we hit record, you were also mentioning that Sam Hoare is going to be there. I can't wait to see Sam again.
No, and he's really excited to go. He got his poster delivered to him this week, and he's like, I don't know if I can show the poster, so I'm going to save that for SLAS, but I'll have it with me if anybody wants to see it.
It's a heavily, it's a rich meeting with a lot of biotech and pharma representatives, and it's interesting because in 2 days we're going to be celebrating the 3rd anniversary of recording the first Dr. GPCR podcast with Paul Insel.
Oh yeah,
and in 3 years, I've got to know so many people, so many companies, so many amazing scientists in the field. And I was looking through the program, and I was like, podcast was a guest, spoke to them, is going to be a guest. And it's just such an amazing, it's like going home.
Yeah. And Paul is such a great person. I really enjoy talking to him whenever I see him in San Diego. And we work a lot. He had a postdoc in his lab who is now, I just found out a couple days ago, I was talking to him on the phone, Ren Ostrom. And he is now the dean of the pharmacology department at Chapman University.
Wow.
And so like he just rose up right through the ranks pretty quick. But he has been so supportive and like was an early adopter of our caddis sensor for cyclic
Mhm.
AMP and did some really neat studies. And it's just really great to when you see that, sort of that tradition being carried on from the labs, people my age who are now starting to retire, like Paul, maybe, and, there are postdocs going on to
Yeah.
do great things. So that's really one of the fun things about this field, isn't it?
I think so too. I think so too. Speaking of the podcast, so this month, February, well, yes, the entire month of February was dedicating to Dr. Mark Caron. We've spoken, as I had mentioned, to over 30 scientists. Next month, we're going back to our somewhat regular programming. So I'm excited to share that we're going to have Dr. And I'm having trouble with the name, and he gave me a trick on how to pronounce his name. So his name is Raul Gaitnedinov. He was actually working for a long time in Dr. Caron's lab, back and forth. I think he has an amazing story to tell about his time in the Caron lab, but also his personal story and philosophy. He's a dopamine person since a young age, and people are going to be able to really learn more about his research. I'm excited to share that Annabelle Milner is also going to be a guest on the podcast. She is a PhD student at Imperial College London in Eileen Hanyalolu's lab. And I emailed her and I said, Annabelle, do you want to talk to me? And she said, yes, which I have to say, I have to applaud because a lot of PhD students and a lot of postdocs sometimes feel like with the caliber of the guests we have on the podcast, they don't have enough to contribute. Well, if you listen to Annabelle's episode, you'll see that anyone who is in the GPCR field has contributions that they can talk about on the podcast.
Oh, yeah, that's so true. And not only that, is all those big dudes, those guys with
Yes.
the Nobel Prizes and everything, they used to be postdocs too. So
Yeah, exactly.
they were, believe it or not, they were young at one time. And I know
Exactly.
you had Scott on the
Yeah.
podcast at one point from Kinetics, and
Yeah.
I was just laughing because he's such a big person now in the field, and just remembering him from Wiley Vale's lab as a graduate student at Salk back in the day. So.
I love talking to him, and he was very humble. I really enjoyed talking to him.
Really
Amazing,
nice guy.
amazing episode. Yeah,
Yeah.
yeah, yeah. The other two guests in March are going to be Dr. Bruno Giros, also a former Caron lab postdoc, currently at McGill University. We had a great talk again about dopamine, but I think dopamine never gets old, especially talking to someone who has a neuroscience background. And then we're going to close the month of March with Dr. Roger Sunahara. And I recorded the episode with him just days before the surprise symposium that was organized by Brian Kobilka at UCSD for Roger's birthday.
Nice. Very timely. That's very timely.
Yes, yes, absolutely. Very timely. And I think people are going to love hearing his story and learning that he is actually an instrument maker.
Oh,
And
musical?
yes. Yes.
Oh, very cool.
Yes. And that's all I'm going to say because I want people to wait and listen to it. Obviously, Ecosystem Premium members are going to have access to all 4 episodes with the videos ahead of time before these get released. But I think, all of our guests for the next month have very unique stories, as everyone else does.
Yeah. I've been thinking about that lately because the theme of our booth this year at SLAS is the song of the cell,
Mm-hmm.
and so we tried to kind of incorporate that idea of the symphony that goes on within the living cell,
Mm-hmm.
and how complex and beautiful and trying to express our biosensors as tools that allow you to experience that music of the cell. And it all came down because I was reading Siddhartha Mukherjee's book, and he's such a good writer, and so so expressive about biology,
Yeah.
and it sort of inspired our booth. So I'll send you pictures.
Great. I can't wait. Yeah, I saw Sid give it was more of a discussion at Rockefeller University when his I think and one of his books came out, and he's such a charismatic person, and he's such an amazing and elegant speaker.
Yeah.
Absolutely worth reading the books.
Oh, yeah. They're so good.
The other thing I wanted to mention in the episode with Roger Sunahara, people are going to find out who in the field is actually a great musician.
Oh, nice.
I'm not gonna spill the beans. I want people to wait. Maybe when we stop recording, I'm gonna tell you, Anne Marie. But I want people to to come in and listen in, and you're gonna be. I mean, your jaw is gonna drop. I think it's gonna be a great surprise. I want to also mention that ecosystem premium memberships are for everyone, but if people are having financial difficulties or live in a developing country, just email us at hello@drgpcr.com and we're going to help you get into the ecosystem. And last but not least, I don't know if you've noticed, and I don't know how much you like them, we have started classifying our GPCR news and sending out weekly updates with fewer articles because we feel like it's easier to digest just a couple of articles.
I like that. I like that idea. You can just enjoy it and then get back to
Yeah.
your thing. You don't want the New York Times. You just want, yeah, a little treat.
Exactly. And I'm glad you're saying that. I just also want to mention that you can adjust your email settings. So when the news come out on a weekly basis, they're scheduled to come out every Thursday at 10 a.m. Eastern time. But if it's too much, then there's a way to adjust in the ecosystem the email frequency from 0 to as many as you'd like. And last but not least, I want to highlight our newest partner, Orion Biotechnology's white paper on their platform that they just published in the ecosystem. I can't wait for you guys at Montana Molecular to have your contributor account and to start putting out their scientific content to our members. And
Yeah, well, we have so much, and we just want to
yes,
share this. Our whole mission is to try to share tools with people and share ideas, and so to have um, a specialized place where we can do that. Of course, we can do it on our website. Of course, we
yeah,
can. But this is a more like a community space, and so
yes,
I really like that.
yes.
We'll be
Speaking
sharing.
of sharing, we just learned not too long ago that now ecosystem members get a discount at Montana Molecular with a coupon code.
That's
It's
right.
not it's not yet implemented, but it will be very soon, hopefully in the next couple of weeks, 2 or 3 weeks. And then people can place orders at Montana Molecular and use the Dr. GPCR coupon code.
Yeah. And if they're in a big hurry in the next 2 to 3 weeks to get some experiments done with biosensors, just tell us. We're a small company. We know each other. So you can just tell us. We know you, Mina.
And I know Anne Marie, so feel free to just email, to send an email and then mention the ecosystem to get that discount. I cannot remember. I think it was a 15%
That sounds about
discount,
right. Yeah.
but
That's,
don't quote me on that. I'll have to verify.
no, I think that sounds right. And yeah, we're here to help. So we want
Yeah.
people to be able to, just like you, we want people to be able to use these tools that we're developing, because really the ecosystem is a tool for people,
Yes.
to use and to make life easier.
You make a great point, and I think our goal for the next year or so is to show off what the ecosystem can do. There's opportunities to interact in the ecosystem. We have groups, forums. Soon the Montana Molecular Group will be up and running. So if anyone has any questions, they can direct them to you into the group, and they can also scroll back and see if maybe they can find the answer to their question. Because it's like Google. You type in a question, and by the time you finish the first word, it already completes the sentence because you're not the only one who searched for that term.
Yeah.
All right. I think that was the news on our end. Was there anything else on your end, and Marie, that you wanted to share?
Well, let me think about that. I mean, we're well known for our work with the GPCR biosensors, um, for signaling, and we also have some other projects going on, that are new that we haven't really, um, put out there yet because they're
Hmm.
still under development. But we are working on a new sensor for row A that we hope will be a way that people can look at G1213 signaling. So we have a couple of GPCRs that are, they're, I think that that's their main, pathway of signaling, and we can get a little calcium response, but we really want to see, if this row A sensor will be a good, way to look at G1213. And we've got some GPCRs that, we are known to signal that way. And the other technology we're always working on refining is our gene delivery for, the BACMAM system. So
Hmm.
all of our biosensors, you make biosensors in, and it's like, what's the best way to deliver that gene into the cell?
Yeah.
So, um, there was a baculovirus-based system called BACMAM that was developed, I don't know, 20, 25 years ago, that was used a lot at GSK.
Hmm.
And we've been refining that and improving it over the years and have different variations now. And one of the things we developed in the last year was, well, what we loved, I'll back up a little bit. What we loved about Bachman was you could very quickly scale up a large quantity of the virus in SF9 cells, right?
Yes.
And then, once you put it in a mammalian cell, it didn't replicate or integrate or anything. So it was nice for a transient kind of expression. But the expression looked so consistent, more like a stable cell line.
Hmm.
And it um and we just really liked that consistency and the ability to control the level of expression. So that became really important when we were developing the Borealis, sensor for arresting, because the amount of receptor really could
Matters.
matter, right?
Yeah.
So, we're doing all this optimization with the BACMAM, and then we had a customer approach us and ask us, "Hey, could you, instead of making a lot of one, virus, you know, carrying, say, a biosensor, make up, 200 ml or something, instead of a lot of one thing, can you make just a little bit of a whole bunch of different genes and put it on an echo plate?"
Hmm.
And that kind of automation was really engineering intensive. There was a lot of work that went into that. But now we've done it. And so now we can pretty easily do these kind of, automated, I'm going to call it a high throughput,
Hmm.
um, kind of application, even though it's not necessarily high throughput screening. It's more like,
Yeah.
what if there were, say, a GPCR that had, um, so many different known, point mutations that affected the response of the drug? So let's just say it's, the MC4 receptor or something like
Yeah.
that. And there's all these different known mutations that have different profiles. Can we, in a 96 or 384 well plate, just make all those different mutations in a virus and then very quickly, look at how that changes the signaling profile with these changes
Wow.
in the receptor? So, and or another application would be, of course, like putting a guide RNA and a Cas9, say, in the
Yeah.
cell, or at the same
Yeah.
time for a
Yeah.
knockout. So we're excited about that application and just starting to use it to screen for, in our evolution of the sensors. We make thousands of different versions of the sensor, and then we screen for brightness and for signal, right?
Yeah.
So now we're using it to do that just because we only need a little bit, you know?
Exactly.
And having it in the backmam means that we can put it in a cell, really any cell, and then see how it performs, with the virus, rather than having to deal with like a lipofectamine-based transfection, which
Yeah.
is expensive and variable and all those kind of things.
Yeah.
So
I think it's very neat.
yeah,
It's very neat.
so those are, those are the things that we're working on that are not biosensors, and then related to the biosensors. One thing we did this year was to evolve a brighter, faster folding red fluorescent protein. So one of the big problems with the reds is that they are slow to fold and they're much dimmer than the greens.
Yeah.
And in vivo neuronal applications, they tend to clump.
Okay.
And so we really wanted to put some effort into getting a better red. Now we haven't tried it in vivo yet, so that's coming. But at the annual biophysics meeting next week, we'll be showing the first results of that red fluorescent protein that we hope we'll be able to make new biosensors with a faster folding, brighter red very soon. So that's on the horizon.
That's very exciting. I can't wait to see the data.
Yeah, we're busy.
We were just complaining about it before we hit record.
Yeah. And we're small, but we're very mighty.
Yes, I know. You were talking in my mind. I was like, okay, I know I understand the business that you were talking about because you guys have all of these projects up and running.
Yeah, we love science. That's the problem with us.
Right? I know. I know. Amazing. All right, Anne-Marie, thank you so much for joining me for this February edition of the Dr. GPCR newsletter. Well, actually, news video. I'm still trying to figure out what's the best nomenclature for this. But I will see you in a couple of minutes after I stop recording. And I hope everybody enjoyed learning these new things, both about Montana Molecular, about our upcoming events at Dr. GPCR.
Yeah. And
All
go
right.
see Sam if you're at that GPCR summit. Go meet Sam. He's the best.
He is. I can't wait to see him. I haven't seen him since May of last year. I'm excited to see him again. All right. Thanks, Anne-Marie.
Thank you, Yamina.